This is a production of Cornell University.
Thank you so much for the introduction, and I would like to thank you guys for having me present my research to you through this virtual visit. And so I'm at the University of Washington-Bothell, and I like to always just give a little bit of context about the institution that I currently work for, because most people are usually very curious about this. So the University of Washington has 3 campuses, and many of you would be familiar with the Seattle campus, which sometimes is described as our flagship campus. But there are 2 other campuses, one in Bothell, where I am, and the other in Tacoma. In terms of the proximity, the Bothell campus, we're about 15 miles away from the Seattle campus, which in reality seems close, but in Seattle traffic, that could actually really be about an hour of your time. And so this is a table with some data from enrollment in fall of 2020. I apologize, I didn't update it for this talk. But what you can tell from this table is that the University of Washington-Bothell makes up about 10% of the total UW enrollment. And when you look at the percentages of student populations, you would also notice that UW-Bothell and University of Washington-Tacoma are both considered primarily undergraduate institutions. And indeed, these campuses were established to provide more access to undergraduate education in the Seattle area or in Western Washington. And so, like most primarily undergraduate institutions, we have a relatively high teaching load. And so in a position like myself, as a tenure-track faculty member, I teach about 5 full courses an academic year. And yes, I always have to say full courses because, not short modules within a course. And so that is quite time intensive as well. And another thing to take into consideration is that as a primarily undergraduate institution, undergraduates are the people in the lab. In my degree program, biological sciences, for example, we do not have a graduate program. So we don't have master's students or PhD students in our program. And so undergrads are in the lab, which is actually presents really exciting opportunities for them and also for me. But as we all remember ourselves as being undergrads in the lab, it is a lot of learning. The learning curve is really steep. And then the students tend to graduate right after they become really, really efficient. But I've really enjoyed working with my undergraduate students and who have contributed to my research program. And so my research interests actually stem from my own upbringing. I was born and raised in the southern African country of Zimbabwe. And in Zimbabwe, we love our maize. We love our corn. It is a staple. It's one of our staple crops. And we use it to make sadza, which is shown here. And so it's a paste that's a little bit more consistent than grits, for example. And so sadza is a staple for Zimbabwean families, and we consume sadza multiple times a day. But like most African countries or Southern African countries, Zimbabwe is subject to recurring droughts. And I remember as a child, this one specific drought season that I specifically remember when, our livelihoods were really devastated. And, these recurring droughts not only affect human livelihood, but also livestock and so forth. And so it is my desire to understand the mechanisms of how we can improve crop tolerance to environmental stresses such as drought. And so we're all aware of the importance of maize as an agronomical crop plant. And so this is an image from I went to a field nursery when Juana Diaz, Puerto Rico, when I was a postdoc. And so maize has a lot of uses that range from animal feed, a source of fuel and food. And the United States is the leading exporter and producer of maize products around the world. And so has really important economical impacts as well. And so I know this figure is a little bit outdated. This graph is a little bit outdated, but we're looking at corn production in the United States over a 10-year period. And one thing that we notice here is that you have this gradual, what appears to be a gradual increase in production over time. But you can't help but notice this dip that happened in 2012 that corresponds to the 2012 drought season. And this was another reminder that abiotic stresses dramatically affect productivity and yield. And this could be different types of stresses. And in this case, we're talking about drought. So again, just like the things I experienced in my childhood, this was a reminder that the United States that I now call home is not immune to these challenges as well. And so we can't, in talking about maize, and maize is a model organism for studies, we also cannot separate ourselves from transposable elements that comprise a large part of the maize genome. And so in this graph, we're looking at percent of TEs in multiple different plant species. And so I'll try to direct your eyes to the very end here, where we're looking at maize. And one thing you notice is that maize, and many of you will be familiar with this, that the maize genome is comprised of about 85% transposable elements. And I think it was, so in our community, we are actively thinking about how the genome and the transposons, the protein coding genes, and transposons affect and interact with each other. Indeed, maize was a pioneering model for studies in epigenetics. And one of our favorite people, Barbara McClintock, made pioneering discoveries in this area. And so maize has been used as a model in genomic imprinting, the studies of pyramidation. And what is relevant to my research and my talk is how transposable elements were first identified in this system, and understanding how they're inactivated through DNA methylation. And so in terms of epigenetic modifications that regulate or affect transcription, there's multiple types that could be modifications to DNA, chromatin modifications, and non-coding RNAs and RNA modifications. For the purposes of my talk, I'm specifically focusing on DNA modifications, specifically cytosine methylation, so methylation of the cytosine base. And so these are modifications that are superimposed onto genetic information that affect gene expression and ultimately affect phenotype. And so plants have evolved two additional, and now we know there's more, DNA-dependent RNA polymerases. And I'm specifically interested in RNA polymerase 4 and RNA polymerase 5. And so they are related to POL2. And these function in a plant-specific epigenetic regulatory pathway that functions specifically for transcriptional silencing. And indeed, in maize, there are additional subtypes of these POL4 and POL5, which increases the complexities in studying these mechanisms through using genetic studies, as there's likely redundancy that goes on. Okay, and so POL4 and POL5 maintain genome stability via an RNA-directed DNA methylation pathway, also known as RDDM. And this pathway is reliant on the biogenesis and response to small interfering RNAs. And so this is a beautiful model that, from a few years ago that was derived from Arabidopsis that came from many years of biochemical and genetic analyses. So this model is pretty complex, and I don't intend for you to grasp everything in detail, but basically it begins with pol-4 dependent biogenesis of small RNAs that are processed and incorporated into a complex that involves pol-5, and this mediates chromatin alterations and also de novo methylation at target sites. So for this pathway, and relevant to my studies, one important protein is RDR2, which is an RNA-dependent RNA polymerase that functions in creating a double-stranded RNA molecule using a single-stranded template that's produced by pol-4. And so this is our very simplified model from maize and the components that we understand from the same pathway in maize. And so one of the genes that I'm really interested in maize is known as MOP1, or mediator of pyrimetation 1, which corresponds to a putative RNA-dependent RNA polymerase. So it's autologous to RDR2 in Arabidopsis. Again, just reminding you that functions to create a double-stranded RNA molecule using this single-stranded template. And so in maize, MOP1 is required for progression of the RDDM pathway that will ultimately induce de novo methylation. And we know that in using MOP1 mutants, we're able to identify physiological consequences of loss of epigenetic regulation through this RDDM mechanism. And so these are some images from the original paper for the identification of MOP1. And in this image here, we're looking at a normal maize tassel, which is the male inflorescence. And in MOP1 mutants, we observe a variety of developmental phenotypes. And so in these other three images, we're looking at mutant phenotypes that were observed that vary, for example. And so this would be described as a feminized tassel, a barren tassel. And when you get one of these mutants in the field, you just completely give up for the day. And so these developmental phenotypes or defects are consistent, but not predictable, right? So in a given growing season, you cannot predict which phenotypes you are going to observe. And they are influenced by the environment. And this is one of the things I noticed as a postdoc, how slight environmental changes would also enhance these mutant phenotypes. Another thing to note is that most of these defects affect floral inflorescences. And you can imagine how that also affects yield or seed yield ultimately. And so this shows us that a MOP1 mediated RNA RDDM pathway, is important for normal growth and development. And so one of the genes that we know is regulated in a MOP1 mediated manner is known as B1. So the B1 gene in maize confers purple pigmentation in specific plant tissues. And so in this example, again, we're looking at the male inflorescence tassel, and we know that, and this is a very oversimplified model, but in MOP1 mediated RDDM pathway, induces methylation of the promoter of the B1 gene, right? Which silences transcription of the B1 gene. However, in MOP1 mutants, we get this purple phenotype because With the loss of MOP1, we are unable to methylate the promoter of the B1 gene, and B1 is then transcriptionally active in tissues where it's not supposed to be expressed. And so this is one example of a very direct MOP1 mediated transcriptional silencing pathway. But over the years, we now know that many genes are silenced either directly or indirectly by a MOP1 mediated mechanism. And previously, we also showed that this mutation at the molecular level disrupts the majority of RDDM. And so we used the MOP1 mutation to characterize regions of the maize genome that were undergoing RDDM. And what we were able to determine is that RDDM loci were within 3 kb of the transcription start site of protein coding genes. We were also able to look at small RNAs and measuring small RNAs. And in this figure, we're looking at small RNAs of different sizes in the MOP1 mutant in blue here, and in the wild type control, which is in red. And we noticed that the mutant, in the mutant, we observed a significant reduction in 24 nucleotide small RNAs of all sizes at regions of the maize genome that were undergoing RDDM. And we also measured cytosine methylation. And so we're looking at percent methylation in different contexts of cytosine methylation, where H is either an A, T, or C. And one thing that we notice here too, that you see in this graph, is that it's a specific context of cytosine methylation, known as methyl CH8, that is affected primarily at genomic loci that are undergoing RDDM, with very little changes in the other contexts of small RNAs. Okay. And so just in summary, we were able to identify that these methylated, what we call CHH islands, are often located at the edges between transposons and highly expressed genes. And so in this cartoon over here, we're looking at a protein coding gene with a transposon upstream of that gene. And we're looking at cytosine methylation or CHH methylation at 4 different positions of that transposon. And we see that the region that's closest in proximity to the gene have more elevated levels of CHH methylation. And so with years of data, we're able to partition the maize genome based on cytosine methylation and what we imagine, or what we understand, excuse me, to be happening in the context of RDDM. And so using a variety of genetic mutants, we know that the maize genome can be partitioned in regions that are euchromatic in white here and heterochromatic in red. And at euchromatic regions, many maize genes are found in proximity to transposable elements, and that a specific context of cytosine methylation functions to establish boundaries between gene proximal transposable elements and these protein coding genes. So at this stage, we had done a lot of work collaboratively to characterize RDDM in the maize genome. And during transitions, there was really great opportunities for you to reflect about what you want to do. And so as I was finishing off my postdoc and transitioning into my own research program, I started to reflect about what my motivations were. And my motivations went back to my own interest in understanding the effects of the molecular mechanisms that contribute to that are affected, in drought responses, in a way that we can overcome those challenges. And so my independent research program, I aim to understand how epigenetic modifications or mechanisms facilitate growth, development, and responses to abiotic stress in agriculturally important crop plants. And I aim to use an integrated approach to identify synergy between two regulatory mechanisms. And in this case, I mean epigenetic mechanisms and the mechanisms that function in responses to abiotic stress. And I aim to understand how these networks contribute to plant growth and development, and specifically in divergent and related members of the grass family, with my ultimate goal of being able to manipulate these responses or mechanisms to improve crop productivity. So for the next few slides, I'm going to just describe some of the work that we've done. And so here's an experimental approach for one of our most recent studies. And so we incorporate molecular genetics in which we can manipulate, perform some sort of genetic or epigenetic manipulation, in this case, using the MOP1 mutant, and subjects this genetic stock to some sort of environmental stimulus and in specific tissues and in specific species. And then so in this case, we're looking at a MOP1 mutant that has been exposed to a stress hormone ABA in seedlings in maize. And so we took seedlings from a segregating family, a heterozygous family that was self-pollinated. And then we identified the homozygous mutant and the homozygous wild type individuals. And so the mutant would be deficient in RDDM, and the wild type individual would have a functional RDDM pathway. And we subjected this to an environmental stimulus. And in this case, we used ABA in this pilot study, and I'll explain why in the next slide. And so this setup allows us to do a multi-level comparison between genotype and treatment. And then we can then further subject this to genomic and bioinformatic analysis. And so just as a refresher, that ABA is a hormone that accumulates in plants under some abiotic stress responses. And so ABA has a variety of, plays a variety of roles in the life of a plant, but is also involved in responses to environmental stress, in which you observe transcriptional activation of stress responsive genes, and also physiological responses to prevent water loss. Again, I want to emphasize that ABA is not involved in all abiotic stress responses, but we did use it as a proxy in this experiment to simulate abiotic stress. And so there are some stress responses that are ABA independent and those that are ABA dependent. Okay, so with our genetic and molecular genetic setup, this allows us then to perform RNA sequencing and bioinformatic analysis to identify differentially expressed genes and differentially methylated regions, for example. And with time, as we increase our sampling in different tissues, we can then also start to create gene regulatory networks and gene co-expression networks. And so in one study, and this is as a collaboration with my postdoc advisor, Karen McGinnis, and we looked at epigenetic regulation of ABA-induced transcriptional responses in maize. And so just to describe some of the results from this study, we used an RNA-seq approach to measure the transcriptional consequences of disrupting these two regulatory networks. Again, an epigenetic network and an abiotic stress responsive network. And from our data in this graph here, we're looking at the number of genes in 4 different pairwise comparisons. And so we identified about 3,200 differently expressed genes in these pairwise comparisons. And so a specific comparison is classified here based on color. So for example, we looked at wild type individuals subjected to ABA relative to control, or we looked at MOP1 mutants not treated with ABA relative to wild type control, and so on and so on. And in each comparison, as I'm showing here, we looked at genes that were upregulated and genes that were downregulated. And so the first column for each color would be genes that were upregulated and genes that were downregulated in each. And so we're able to get different levels of information based on each pairwise comparison. For example, what we observed and what we'd expect is that, like in comparisons in subgroup 1 and 2, which would be wild type ABA over the control, we identified canonical abiotic stress responsive genes with some maize specific variations in a comparison between in groups 3 and 4, for example. This one would just give us information about differentially expressed genes and seedlings, because this was an ABA independent study or comparison. One thing that does stick out the most is that the majority of differentially expressed genes were identified in the comparison when you disrupted RDDM, so loss of MOP1 mutant or loss of RDDM in addition to the stress hormone ABA relative to control. And one of the most interesting categories would be analysis in groups, what we're calling 7 and 8, because this allows us to identify genes that are dependent on RDDM activity under abiotic stress through a comparison where you disrupt some MOP1 mutant subjected to ABA relative to a wild type individual that was subjected to ABA. So we're further characterizing these genes, but just in case people are interested, genes that were enriched in this category were associated with reproduction, nucleotide binding, developmental processes, and transcription factor activity. And so that leads me to this slide, in which we wanted to further characterize the functional categories of genes that were differentially expressed in our analysis. And so we performed a singular enrichment analysis in gene ontology specifically for biological processes. I don't expect you to be able to read all the different subcategories. I'll summarize them for you in a little bit. And so in this image, we're looking at the log 10 of % genes in each category, where a deep red indicates that there were more genes in that category, and a deep blue means that there were fewer genes in that category, but they were genes in that category. And so white or missing means that there were no genes identified in that category for that treatment. And we did this, and we looked at this in ABA treated wild type seedlings and mutant seedlings that were downregulated and those that were upregulated. And so one thing we noticed is consistently response to stress, response to stimulus was common to all, as these had all been treated with ABA. But one thing that you would also notice that sticks out is this unique category that was observed specifically in MOP1 mutants, in which genes were the downregulated category of MOP1 mutants. And these genes specifically associated with cell size, growth, and differentiation, and affecting floral and reproductive development. And so this is consistent with some of these phenotypes that we observe. And so this tells us that in the absence of RDDM or MOP1, ABA induces differential gene expression with distinct functional categories. And again, we're interested in characterizing these genes that fall into these categories in a little bit more detail. As well as transcription factors that we've identified, many transcription factors were differentially expressed in our samples. And about 60% of the differentially expressed transcription factors were unique to the MOP1 mutant. And so transcription factors play key roles as regulators of gene expression, affecting multiple target genes directly or indirectly. And so we looked at the differentially expressed transcription factors in wild type and MOP1 mutants, green for wild type and purple for MOP1 mutants, that were either upregulated or downregulated. Again, in this, we identified some transcription factors that were unique to wild type and some that were common to wild type and mutant, and also those that were unique to the mutant. And again, that the majority of differentially expressed transcription factors were unique to the MOP1 mutant. And so we wanted to probe this a little bit more because this data suggested that that RDDM mediated responses to abiotic stress involve transcription factors to a large extent. And so we took advantage of a model that had been elegantly described by Joe Ecker's group, which is through a really extensive study in which they were able to position transcription factors in a hierarchical network in which a upper level transcription factor, like a level 1 transcription factor, will affect multiple downstream targets, right? Compared to a lower level transcription factor. And so in their study as well, they also used ABA. And so ABA is presumed to then activate a specific type of transcription factor that recognizes specific ABA responsive elements of a level 1 transcription factor, for example. And that then creates another transcription factor protein that activates a level 2 transcription factor, and so on until you have your physiological stress response. And so we identified maize orthologs of the different transcription factors at the different levels. And again, so this would be a level 1, a top tier transcription factor, level 2 transcription factors, and level 3 transcription factors, and then downstream target genes. And so in this image, we're looking at the colors indicate log2 fold change in expression, where a deep red in this case would be highly expressed genes, and deep blue in this case would be highly downregulated genes. And also something to note, if I could draw your attention to the bottom here, is that if you see a solid black circle around that, that means that gene was differentially expressed specifically only in wild type individuals. And a dotted circle means that gene was differently expressed specifically in the MOP1 mutant. And if there's no border around it, that means it was found common to both. And so one of the things you'll notice is that we have more individuals that are, more genes, more genes that had that were unique to the mutant as well. And so then using gene regulatory networks, we're able to make predictions about what could be happening and what transcription factors are affecting which targets in this pathway. And one thing that we also noticed is that we did check to identify that we did identify ABA responsive motifs in the majority of these transcription factors. And so just for the interest of time, I also want I'll really just describe our DNA methylation experiments. And so we did probe DNA methylation using the similar approach, again, using abscisic acid as a proxy for abiotic stress responses in the MOP1 mutant and wild type individuals. In this case, we used genomic DNA that was subjected to bisulfite conversion. But I want to mention that this was just a survey of what was going on for methylation, so not very in-depth. So we used a sequence capture approach that was described by the Springer lab. And so we're only really looking at regions that we know to have been enriched for CHH methylation in the maize genome. So we're only looking about less than 1% of the maize genome. But just to give you a context of what we found, if I'm to use the same color scheme again, where green is wild type and purple is the mutant, we're looking at percent change in methylation in the different contexts, methyl CG, CHG, and CHH. And one thing we notice is that we have an increase in cytosine methylation as a result of ABA treatment in wild type and further loss of methylation in the mutant. And so this shows us that epigenetic mechanisms are really important as a mechanism of response to abiotic stress responses. And as we think about constructing gene regulatory networks, we never, epigenetic modifications aren't always included, but then this tells us that, this gives us some insights of their role in that area. We also looked at transposable elements that were, and the different categories of transposable elements in our RNA-seq samples, excuse me. And we looked at the retrotransposons and also the DNA transposons. And in this image, black indicates the reference genome, which is B73. And then, and the white bars would be MOP1 mutant upregulated genes, and the hashed bars would be the MOP1 downregulated. And one thing the overall message here is that in the mutant, we identify specific categories of DNA transposons that are specific to the mutant. And Jason Lin, who is a postdoc in Karen McGinnis's lab, also looked at the physiological consequences in response to abiotic stress in these mutants. And so in this image, he was looking at, in the y-axis, we're looking at days after sowing at different developmental stages in maize. And there's, just to direct your eyes real quick, the pink lines would be the MOP1 mutant that is either subjected to drought or normally watered. And then the blue would be a B73 individual as well, and the green would be a wild type individual, a MOP1 wild type counterpart. And so in this study, what Jason did is he, after sowing, he watered the maize seedlings until they reached a specific developmental stage, in which he withheld watering, again, subjecting these plants to severe drought treatment until they reached another developmental stage, in which then they were watered normally until they reached maturity. And what we see here is that the MOP1 mutant is delayed in its recovery and after drought treatment, showing the role of this MOP1 mediated RDDM in physiological responses to stress. And so in our studies, we've identified synergy between these two important transcriptional regulatory networks in maize. Interpretation is confounded by the hierarchical nature and cascading effects of transcriptional outcomes that is part of both regulatory networks. And so, because each pathway is dependent on its own sense and trans regulatory elements, for example. And so we did use another time course experiment to identify more direct or primary and indirect regulatory targets. And that paper came out recently. And so characterization of these complex regulatory interactions has translational applications and allows us to be able to manipulate these mechanisms for enhanced stress tolerance in maize and other crop plants. Okay, and with that, I will end here. I'd like to acknowledge several students in my lab who've contributed a lot to the small pond ABA study, including Catherine Liu, who actually was one of the co-authors in our research manuscript. Okay, and I'll take questions at this point.
Okay, let me see if anybody.
I have a question.
Oh, okay.
Does the mutant, the MOP1 mutant, have any phenotype in the early vegetative stage before reproduction?
Did you hear the question?
Yeah, so if I heard the question correctly, does it have a phenotype in the vegetative stage before reproduction?
Yes.
Yes, a little bit later. So at the seeding stage, we don't notice a phenotype, but we start to actually see expression of the B1 gene during at the vegetative stage, and that's one of the ways that we actually can phenotypically identify the mutants. And also, they tend to be much smaller in size and really with really thin stalks.
Thank you.
But at the in some of our studies that we do at the seedling stage, we have to genotype using molecular techniques because we aren't able to distinguish at that stage.
I have a question about working with undergraduates. How do you deal with the high turnover and also still make them feel like they're able to contribute to the research in a real and real way, not just busy work?
Right. So, yeah. So one of the things when I was a postdoc, when I was a senior postdoc, Carrie McGinnis allowed me to design projects for rotation students. And I would design projects that were based on the research that I was doing. And it so happened that rotations at Florida State were about 10 weeks. As life or fate would have it, the quarter system is 10 weeks. How's that for job preparation? And so that was a great opportunity for me to learn how to design projects in short for short term. And the thing about it is just breaking a big idea into smaller chunks.
You also have a long growing cycle for corn. Corn takes a lot longer to grow, so it's not a Arabidopsis. So how does that complicate your study?
Right. So to date, we have been using seedlings, the seedling stage, so 10-day-old seedlings for the studies that we've been doing in my lab. But now that I want to work on the real applications, right, I need to expand a little bit more. And so with that, I am transitioning into Soteria as well. And I'm working with Andre Eveland to try create genetic stocks in Soteria so that we can, which has a little bit shorter life cycle and is more conducive to growing in a growth chamber, for example, and also in the Seattle area. But yes, so far, the seedling stage is what we've been using. And so we haven't really been going full season.
I see there's a question from Ed. Ed, do you want to just get off mute and ask it?
Sure. Yeah, so Thelma, thanks for the great talk. And yeah, I was wondering, do you think you have enough knowledge of the sequences and motifs that you could actually pick up that model and apply it to, say, the NAM parents and just look through the sequence and start giving predictions as to where the ABA responses would change? Yeah,
that would actually be really something exciting to look at. Yeah. So at this point, we have enough data to, to start to make some predictions, and then testing those predictions is really the next level.
All right, there's also a question from Lee Cass in the chat. Where do you grow your maize plants? H. Roman went to Seattle in 1942, but changed his research program to yeast.
Right. So this is where, long term, this is where collaborations are going to be key, right? And fortunately, the maize community has been really supportive of me and, offering for me to, borrow space in their field in the growing seasons. But again, during the, so far, we've been using 10-day-old seedlings that can be grown. We do have a greenhouse, but you also sometimes use a grow chamber.
Thank you, Eric.
But that is not, yeah, it's not sustainable long term for sure.
Eric, you can come off mute if you'd like.
Okay, so I hope you can hear me. Thanks so much. I really was interested in the slide about ABA inducing changes in methylation, and so you may have explained this, and so, but I didn't quite catch how many of those changes were happening in, say, the TE component versus RDDM targets that you'd identified previously, or in the coding sequences.
Right. So one caveat about that methylation study was that those were regions that we already knew to be enriched for CHH methylation. So what we're defining as RDDM targets. And so, so those are mostly enriched for those regions that were between the protein coding gene and the transposon. So those boundary regions.
Okay, great.
So they had already been established to have elevated levels of CHH methylation.
Okay, great. Thanks for the clarification.
Yeah, no problem. Thank you.
Any questions from the audience here? Anybody else on chat that has a question or would like to come off mute to ask a question? Okay. Thelma, I think you're meeting with the individual people throughout the day. Are you meeting with students at some point today?
I've already met with the students, so that was a great meeting. Yeah.
Okay, great. Well, I guess we'll end early and give you a break so that you can get to your next Zoom meeting and have a moment to, get a drink of water or something. Thank you so much for joining us today. I appreciate you being here virtual. It's a very difficult way to meet people and to express yourself and build collaborations, but I'm really glad to have you here and thank you. And hopefully we'll get you here to Cornell sometime in the future. Thank you.
Yes, thank you so much. Yes, you guys do owe me a visit. well, I need to come and visit for sure. Thank you all for attending. I appreciate it.
Applause, please. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.